When we picture clinical infections, it's incredibly easy to just imagine a chaotic, invading army. But to truly understand how these microscopic pathogens actually survive our immune defenses and, our strongest antibiotics, we have to look way past their armor. We really need to examine their internal escalation arc. We're talking about the incredible hidden structures deep inside that act as the ultimate survival engines, driving the exact diseases you're going to encounter in your clinical careers.
So, in our last explainer, we successfully breached the outer fortress of the pathogen, right? We explored the cell envelope, the walls and membranes that act as that primary barrier. But now, we're stepping right past those gates and walking directly into the command center, the bacterial cytoplasm. This is where the actual life or death decisions of the cell are executed.
Okay, let's dive right into our roadmap for this explainer. 1. Inside the bacterial command center. 2. Plasmids and genetic resistance. 3. Internal machinery and structure. 4. Inclusion bodies for storage. 5. The endospore life cycle. And 6. Clinical threat of endospores.
Starting off with Section 1, inside the bacterial command center.
Now, you might think the cytosol is just empty space, but it's absolutely not. It's the fundamental, highly dynamic environment that keeps the cell alive and kicking. If you look at this composition breakdown, it's essentially this dense, gelatinous aqueous gel. It's roughly 70 to 80% water, but honestly, it's that remaining percentage that makes all the difference. This fluid acts as a solvent for a super concentrated solute pool. We're talking a complex mixture of amino acids, sugars, fatty acids, and essential inorganic salts. And floating around right within this dense gel are the massive internal functional components of the cell: the genetic material, the ribosomes, the inclusion bodies, and the cytoskeleton.
Moving on to Section 2, plasmids and genetic resistance.
Now, what's really interesting about this slide is the stark contrast in how bacteria organize their DNA compared to our own cells. Bacteria completely lack a nuclear membrane, so their primary genetic material, the bacterial chromosome, just aggregates in a centralized, uncompartmentalized space called the nucleoid zone. It's this massive, single, continuous strand of circular double-stranded DNA holding all the essential instructions for survival. But right alongside it, you have bacterial plasmids. These are tiny, separate, circular pieces of DNA. They're totally independent of the main chromosome, and, this is key, they are non-essential for basic survival. But while they might be non-essential, they carry some pretty serious protective blueprints, like toxin production or drug resistance genes, which we call R factors.
And this right here leads us to a massive clinical headache. Plasmids replicate autonomously. That means they just copy themselves without even waiting for the main cell to divide. Bacteria can then use these structures, called conjugation pili, to dynamically pass those plasmids directly over to neighboring cells. For those of you heading into healthcare, this is absolutely crucial to understand. This is the exact mechanism by which multi-drug resistance spreads like wildfire across hospital intensive care units. One single bacterium figures out how to beat an antibiotic, slaps that info onto a plasmid, and literally just hands it out to the neighbors.
All right, Section 3, internal machinery and structure.
So, floating in that dense cytoplasmic gel are the massive functional components acting as the everyday workforce of the cell. And a huge part of that workforce is the bacterial ribosome. The number you absolutely have to remember here is 70S. That's the total density rating or sedimentation sizing of bacterial ribosomes. This is a fundamental structural difference from human eukaryotic ribosomes, which are 80S. And because of that structural difference, well, it makes the 70S bacterial ribosome a major pharmacological target for our antibiotics. It basically allows us to shut down the bacteria without harming the patient's own cells, a real game changer.
To break down that 70S ribosome a bit further, it's composed of a specialized matrix that is 60% ribosomal RNA and 40% structural proteins. It's built by clamping together a small 30S subunit and a large 50S subunit. Together, they act as the primary sites of protein synthesis. They're actively reading messenger RNA strands to cross-link amino acids into functional polypeptides. Basically, they are the factories building the proteins that keep the pathogen alive.
But hey, factories need a building to house them, right? And that brings us to the bacterial cytoskeleton, which provides some absolutely crucial architectural support. It's composed of long, polymerized protein fibers, like MreB and FtsZ, that are actually surprisingly analogous to the actin and tubulin you'd find in our own human cells. This cytoskeleton forms a really tight, helical network immediately beneath the cell membrane. And it isn't just holding the cell open, It actively directs the patterns of cell wall synthesis, especially during binary fission when the cell divides. By dictating exactly where the new cell wall is deposited, this architectural network determines the overall physical shape of the bacteria. And definitely keep this concept in the back of your mind, because in our next explainer in this curriculum, we are going to focus entirely on those resulting bacterial shapes, arrangements, and sizes.
Section 4. Inclusion bodies for storage.
So when times are good, bacteria actually shift from basic structural components to a highly specialized survival tactic, hoarding resources in inclusion bodies. These are specialized storage compartments that either lack a membrane entirely or they're enclosed by just a simple single-layered protein shell. And they come in two main flavors. You have organic inclusions, which gather energy-rich polymers like polybeta-hydroxybutyrate or PHB and glycogen spheres. Then you've got inorganic granules like metachromatic granules, also called volatin, which store concentrated crystals of phosphate. The cell desperately needs that phosphate for ATP energy synthesis. Both of these types are vital for preventing starvation when nutrients suddenly run short.
But there is a totally genius mechanical reason for storing food this way. So the crucial point here is osmotic protection. Imagine if a bacterium just left thousands of individual nutrient molecules floating freely in its cytoplasm. That incredibly high solute concentration would act like a giant magnet for water. Water would literally rush into the cell due to osmosis, building up pressure until the cell exploded. That's a catastrophic process called cell lysis. So by condensing those thousands of molecules into a few large and soluble crystalline clumps inside an inclusion body, the cell drastically reduces the number of free dissolved particles. It essentially saves itself from popping like a water balloon. Just a brilliant packing strategy.
Which brings us to Section 5, the endospore life cycle.
So what happens when the nutrients run out entirely? Well, for specific genera of bacteria, like Bacillus and Clostridium, they hit the ultimate panic button. They shift from a vulnerable active vegetative cell to a completely dormant endospore. This shift is called sporulation, and it basically plays out like a high-stakes tactical retreat in 4 steps. Step 1, chromosome replication. The cell duplicates its DNA, pushing the copy to one end. Step 2, spore septation. The plasma membrane infolds, walling off that DNA and a little cytoplasm into a compartment called the forespore. Step 3, cortex deposition. The parent cell actually engulfs this forespore, wrapping it up in a heavily armored layer of specialized peptidoglycan and calcium dipicolinate. And finally, step 4, lysis and release. The parent mother cell sacrifices itself, lysing open to release the mature, dehydrated, metabolically inert endospore right out into the harsh environment.
And that endospore, it will just sit there, totally dormant, just waiting. When favorable environmental cues finally return, like, say, a sudden availability of water, amino acids, or specific salts, the germination process kicks off. It's essentially a highly coordinated ambush. The outer protein coat absorbs water, swells up, and cracks open. Boom! The dormant metabolic enzymes instantly reactivate. Get this, fun fact. Endospores extracted from the guts of extinct bees trapped in 25-million-year-old amber have literally been successfully germinated in labs. No way, right? And from the ruins of that spore casing, a fully functional vegetative cell emerges, immediately ready to resume active growth, binary fission, and, very often, the production of toxins.
Finally, Section 6, Clinical Threat of Endospores.
And this brilliantly illustrates why understanding these cellular structures is absolutely critical for your future medical careers. Endospores possess extreme biochemical defenses. That heavily dehydrated core, along with that high concentration of calcium dipicolinate, heavily insulates the internal proteins in DNA. It actually prevents thermal denaturation to the point where they can easily survive boiling water. And on the outside, their thick, keratin-like protein coat completely prevents the penetration of toxic disinfectants, heavy alcohols, and even ultraviolet rays. These defenses are the exact reason why standard sanitation protocols on hospital floors so often fail to eradicate them.
Because of this mechanism, endospore-forming bacteria represent some of the most heavyweight pathogens you will ever face in the clinic. We are talking about the primary causative agents of some severely life-threatening diseases. You've got Bacillus anthracis, which causes anthrax, Clostridium tetani, causing tetanus, Clostridium botulinum, causing botulism, and Clostridio difficile, which causes pseudomembranous colitis, infamous in healthcare settings as the highly contagious C. diff. The terrifying reality is that these endospores can literally survive for decades in dry hospital dust, completely unfazed by standard cleaning.
So, as you progress in your training and eventually step out onto the clinical floor, the big question you must constantly ask yourself is, how will you adapt your clinical sanitization methods and your infection control protocols to fight a practically immortal enemy? I want you to really keep that question in mind as we move forward in the curriculum. Next up, we'll be exploring the shapes and structural arrangements of these incredibly resilient organisms. See you then.